All right. Howdy. For those of you that don't know me, but I think all of you do, my name is Raymond, and my project focuses on the population genetics of loss of function mutations. So let's dive right on in. Quick overview. First, I'll talk about the redundancy of plant genomes, and then a little bit about loss of function mutations. And then I'll go into about constructing a bioinformatic tool to study loss of function mutation frequency, and then move on to how we can apply our tool and how it can be useful to others. So here we have a phylogenetic tree of plants. It's very big and there's a lot of plants, right? Something really nice about this tree is that it has every whole duplication event of a genome labeled with a star. Now, if we were to go down this tree all the way into the angiosperms, we would find our organism of choice, Arabidopsis. Now, starting in the middle is the oldest for the phylogenetic tree. And so if we were to go back up, we'd follow the Arabidopsis ancestry. And if we went on our way back to the origin, and if we were to count all the duplication events, we would get Arabidopsis to be approximately 48 ploid. Now, for those of you that have worked with Arabidopsis, we know that's not the case. As it turns out, there's some evolutionary mechanisms that return Arabidopsis to a diploid state post whole genome duplication. This normally happens thousands to millions of years, over the course of thousands of millions of years after a duplication event. But during this return to a diploid state, not all of the information that we gained from a whole genome duplication is lost. There's a lot of extra stuff still left over. A lot of the extra stuff is non-functional, hence loss of function mutations. But what we're really interested in is why is there even a return to a diploid state? Well, recently, a paper from Dr. Gray Monroe at UC Davis suggests that adaptation through pseudogenization is possible. Pseudogenization is the development of these loss of function mutations after a whole genome duplication event. So a lot of that extra information. In his recent paper, as seen in the figure that I pulled from it, it shows loss of function alleles at genes associated with flowering time and drought adaptation in higher frequency than those without. So this suggests that there's a positive selection for these loss of function mutations. And that's really interesting. Why is that happening? Well, that brings you to the main question for my project is, do loss of function mutations contribute to adaptation of highly redundant plant genomes? So our project has a couple of aims. First, I'll talk about them overall, and then I'll dive into them more specifically. But first, we want to use publicly available software to identify variants in the 1001 Arabidopsis Genome Project. And then after that, we can then begin to construct a bioinformatic tool to estimate the total site frequency spectrum of loss of function mutations. But why are we using this 1001 Genome Project? What is that? Well, first, why are we using the Arabidopsis anyways? Well, it's a really great model organism. If we want to dive into some wet lab work with it, we can. It's really easy to grow, and the genome is really well known. But this 1001 Genome Project, essentially they constructed a genome from 1001 accessions of Arabidopsis. So we have a really good population size to really work with because we're looking at selection forces. And if we want to look at selection forces, we need to look at a population level, not individual. Additionally, the whole genome sequence, there's a lot of variation, so that just really is ideal for loss of function research. Aim one. There's a really excellent software called SNPF. I mean, I say excellent as in it's finicky at times, but pretty much what it goes through is goes through a genome and it will annotate variants in the genome. Now, when we use this software, it also produces something called a site frequency spectrum. As seen here, this is just an example one, not one that we've generated. I just kind of pulled it off the internet, but pretty much it can show you allele frequency from individual all the way up to a population level. So if, for example, at the right hand of the graph, we saw an uptick in the allele frequency, that would suggest a positive selection. And then if there was nothing, pretty much nothing at the end of that frequency spectrum, then negative selection. And then something more like this is more neutral. Yeah, so this is pretty much what we've done so far. This is pretty much a list of all the variants that SNP-F has pulled from the genome. There's a lot, a whole bunch, but we can, what we know about loss of function mutations, we can pull out the ones that are likely to be loss of function. For example, a gain of a stop codon that just stopped the genome, no more protein. It's pretty much kind of dunzos. Stop loss is also likely. If we lose a stop codon, then we have really long sequence, and then we have a lot of amino acids and a lot of biochemical forces that are just, we don't know if it'll even do anything. And then a frame shift variant, or if we move our reading frame, then we're losing potential amino acids that contribute to protein structure. Additionally, when we ran this, we got a site frequency spectra of all variants. So loss of function are clumped up within this as well. And as we see, there's a lot of like negative selection on this graph. However, there's a lot of ways to break a gene. There's 3 that I just listed now. And for example, say we have 2 copies of this, they're the same gene. If we broke them in 2 different places, they would both be loss of function. Their phenotype expressed would be exactly the same, but they would have different genotypes. And this is something we want to take into account. How do we account for this? Well, that brings us to the need for this bioinformatic tool in AIM-2. So essentially what we want this bioinformatic tool to do is combine all loss of function mutation alleles at a single gene into a functional allele. And with this allele, we would then estimate site frequency spectrum, the site frequency spectrum. We plan on doing this in both R and Python, as these are most accessible to most biologists who would be interested in this kind of research. And luckily, from that paper from UC Davis, Dr. Monroe basically went in and confirmed certain LOF mutations that are being positively selected by hand. So we can check our tool with his work, and then if something doesn't pan out, then we go back and try to figure out what's wrong with the tool and just keep on going through that until it starts working, and then we can start adapting the tool for other species, because recently a lot of pangenomes have been coming about, especially in things like maize and sorghum. And if we're looking, this will be really helpful for studying things like loss of function in a pangenome. So summary, we're just getting the project started, but so far we've accomplished annotating the 1001 Arabidopsis genome with the variants in Arabidopsis genome, and we find out how to begin constructing a bioinformatic tool. So next, we need to isolate the LOF mutations from the SNP-F output, and then we can construct and check the validity of our tool. Future directions, we can apply this to other crop transcripts, just like maize, corn, cotton's another big one. And then we have access to an Oxford nanopore. So if we wanted to take this into the lab and test it ourselves with actual Arabidopsis, we can form cDNA libraries and see to what extent these loss of function mutations are being expressed in the lab. Just some references, and I just want to acknowledge Dr. Charborough for advising me on this project, the Cytonuclear Lab for just generally being around and helping me out, and Dr. Gray Monroe is likely to be a future collaborator, and his past work has helped out a lot. RMAC Summit, it's for essentially letting me be able to run all this intensive program on a supercomputer in Colorado, and then Tech for hosting my research, and NSF for funding me as well. Thank you. Are there any questions? Yeah.
I'm kind of unfamiliar with the term pan-genome.
Oh, yeah. Pretty much they construct the genome from similar to with the 1100 Rapidopsis project. They just like a bunch of accessions, so they have a genome constructed from a population.
Okay.
Yeah.
And this one, what was that about the transcripts?
Pretty much applying what we're doing with the Arabidopsis to the to other plants, more economically important plants like corn and maize.
Their RNA?
They're the DNA from the pan genomes.
I saw transcripts.
Oh.
About mRNA.
Yeah.
In the like table with your counts and everything
Mm-hmm.
with those guys, why do you have a couple on red? Is that just because they have high counts?
Let me go back to that.
Yeah, that guy.
Yeah, I believe it's for the high counts. This is something when it runs to Beth, it provides an HTML file, and
Okay.
that's just kind of how it formatted it.
Okay.
So first, great job, Raymond. It's really cool stuff. I was kind of just wondering, when you're talking about you referenced the Monroe paper a couple of times. Are you using that as like validation for your bioinformatic model, or like training your code to match that phylogeny? Is that what the purpose is?
Yeah, so if we're making the code, we can run it on like Arabidopsis and then check to see if it checks out with the paper as well. So I guess, yeah, pretty much.
Hey, I was just going to chime in. I don't have any questions, and I don't mean to stop the audience from asking any, but I just wanted to, this is Faye from NCGR, and for those of you that are interested in pangenomes and pangenomic analysis, we're going to offer our first pangenomics bioinformatics course, and that's February 28th. It's for a whole week. And you know, as you mentioned, it's like, you know, there's all these quality genome assemblies within a single species. And so then how do we manage that? And so you want to remove reference bias and you want to elucidate biological signals at this comprehensive population scale. And so this workshop, you know, people are interested, of course, is, you know, what a pangenome is, how to build one, and then how to perform fundamental bioinformatics analysis on pangenomics data. So just wanted to do a plug on that since you brought it up. Thanks.
Awesome. Thank you. Dr. Stechna?
Yeah, thank you, Faye. And I've got a couple of questions. They're more of a philosophical question, so feel free to elaborate or philosophize. First one is you sort of dismiss, and I don't mean that in a negative way, but you dismiss introduction of a stop codon as leading to the loss of function, and that makes sense, right? But it could also lead to acquisition of a new function altogether. So it could be a source of diversification and elaboration of valuable traits of a genome. Is anybody looking at that?
Yeah, I think just last week even I was talking to Dr. Charbrough about like how can we verify that these are all loss of function, and I don't remember exactly how to. I am sure we ought to verify them. I'm not exactly sure if you want to leave more credence to that, Dr. Charbrough.
Well, I think it's a great question, and like what gets at what is pseudogenization versus some other output like neo-functionalization.
Yeah.
It's I think the bottom line is that it's really hard to study neofunctionalization because sometimes genes can be pseudogenized and then neofunctionalized. So in many ways, we use the term pseudogenization as a simplification of the process. And so we can see like this protein has a premature stop codon, so it's definitely not doing the same thing that it was doing before. Whether that's a new function or not, I think remains to be seen. And yet there are a lot of people that do study neofunctionalization, but it's really hard. And so nobody comes out with new stuff very quickly. But yeah, I'm hopeful that at the end of this, Raymond we can identify, some of these Arabidopsis accessions with loss of function mutations in them, and then grow them and order them and grow them in the lab and extract their RNA and see what's going on. See, are they expressing these, quote unquote pseudogenized transcripts, or are they not expressing any transcripts at all from those loci, or are those even maybe getting into the proteomics a little bit would be even cooler, and see, are there gene products that we don't know about that are stemming from these sort of pseudogenized products, or, all of that stuff. In general, pseudogenes can play a role at the transcriptional regulation level of acting as antisense RNAs, or even as sense RNA regulators, that sort of thing. So there is precedent for pseudogenes being functional. And so we just use this term pseudogenization or loss of function as a simplification of understanding it. But I think you're dead on that it's a process that can sort of swing in and out of function depending on the time frame at which you're looking.
And the function, yeah, thank you very much for this elaboration. I imagine that looking backwards, finding a protein that's really interesting, maybe, an adhesion or an antibody molecule, and then tracking it back evolutionarily into something that used to be something else, perhaps in a longer incarnation, is likely to be even more fruitful than looking from the other direction. I don't know that we have time for the second question, and maybe there is no answer, but why is being diploid so special? It's not monoploid, it's not triploid. Why is diploid so very special, universally special, right? Let's see if your student can address that first.
Yeah. Why is diploid so special? Why is there always a return? It's a really excellent question. And I hope maybe this angle of loss of function mutations can help answer that, because there's always this constant return. I'm not sure if there's an actual reason why already, but that's the best I can think of now.
Thanks.
Yeah, we don't have an answer. I mean, I think it's a central question in evolutionary biology is why diploidy? One potential spot is because meiosis. Meiosis is the best mechanism of getting the exact quantity of DNA into the offspring that has ever been evolved. Meiosis is the best at it. And it works in pairs, right? And so meiosis as sort of a functional policeman of ploidy maybe is the reason that it goes back to diploidy so commonly, is that meiosis is so critical to accurately transmitting the genome, and meiosis works in pairing. So like there's some base level that below diploidy doesn't, you can't take advantage of meiosis as effectively, and above diploidy it just goes back to I don't know. I don't know. It's a good question. I think an important one that we need. That's the forest through the trees. Is why is diploidy out there? Why is it so common? We don't know.
Well, thank you both, and very much. Very enlightening.
Thank you. Are there any more questions? All right. Thank you.
Thanks, Raymond.